Dear friends, very good morning to all. Myself, Dr. G. Sevelango, Assistant Professor, PG and Research Department of Biochemistry, Sacred Heart College Autonomous, Tirupattur. Today we can discuss about isolation and purification of enzymes. Introduction. Enzymes are found distributed widely in the living system and are a major factor in the growth, maintenance and propagation of living system. Wellstrater and colleagues in 1920 carried out the early attempts for purification of enzymes, and the first crystallized enzyme is urease in 1926 by Sumner. Next, need of isolation of enzyme. Record for study about the various aspects of that enzyme. Next, record for researches and as diagnostic component. They employed in forensics, food processing, fermentation and industrial applications, and also required for leaching of ores. Next one, steps in enzyme isolation. First one, selection of source of material. Choose an appropriate source based on enzyme availability and ease of extraction. Sources are animal tissues, plant extracts and microbial culture. These three are major sources of enzymes. First we will see about animal tissue. The tissue is removed soon after the death of animal and kept in cold condition. Next, the homogenization is done in an isotonic medium which disturbs the plasma membrane of organ. In this procedure, the cell organelles are not affected. For disturbing the intracellular organelles, the following methods are used. First one, freezing and thawa. Freezing and thawa means we are cooling, immediately we can heat it. In this condition, cells are disturbed. Next one, sonication. High frequency sound waves are passed to the solution which contains the cell. Next one, drying in vacuum condition. These are the method which disturb the cells. Next one, plant sources, plant tissue. In plants itself, depending upon the type of the plant, the procedures varies. If the plant is a cereal, means the enzymes are extracted from the flour simply by placing in an aqueous medium and stirring it. Next, it is a fruit by mincing and pressing out the juice through a cheesecloth. If the material is a plant leaf or dried seed, first grind in a mortar, then using a festal homogenizer using a suitable solution. In the case of leaves, acetone is used as a solution. This may contain other components like chlorophyll, fiber, etc. They should be removed. Next one is microorganism as a source. The cells of the microorganisms are very strong compared to plant and also animal cells. Therefore, the cell wall is mainly made up of peptidoglycan. Usually two methods are employed. We are isolating the enzyme source from microorganism. First one is drying and another one is mechanical disruption. Both the methods are used. Second one step, extraction. Extraction means cell disruption. Two types of enzymes present. One is extracellular enzyme, another one is intracellular enzyme. First one, extracellular enzyme, easily collected from culture media. Example, phenylamylase. Next one is intracellular enzymes, require cell disruption to release the enzymes. Next, disturbing methods, cell disturbing methods. First one, mechanical method. In the mechanical method, first one, homogenization. Homogenization means breaking cells using the high pressure. Next one, sonication method, uses the ultrasound waves to disturb the cells. Next one, bead milling. Bead milling means grinding cells with small beads. Next one method, chemical method. Under the chemical methods, osmotic chalk. Osmotic chalk means we are used for bacterial cells especially. Next one, we are using the detergents which breaking the lipid membrane of cells, especially Triton X100 used as a detergent. Third one, enzymatic lysis. Enzymes especially lysozyme which breaks the cell walls. Next, cellulase which breaks the plant cell wall. Lysosome for bacterial cell wall, cellulase for plant cell wall. Next, removal of debris, clarification procedure. Removal of debris means we are using the centrifugation and also filtration procedure. Centrifugation means spins the sample at high speed to remove cell debris and collect the supernatant which contains the enzymes. Next one, filtration, uses the membranes filters to remove the solid particles which from the solid particle with the help of filter we can collect the supernatant. Next one, concentration of enzyme. Purpose, concentration of enzyme means we can increase the enzyme concentration for further purification procedure. Here we are using some methods, ammonium sulfate precipitation which is called salting out, separates the enzyme based on solubility. Next one, organic solvent precipitation. Here organic solvents used for acetone and also ethanol which removes the unwanted proteins. Then ultrafiltration, in this condition uses for semi-permeable membrane to concentrate the enzymes. In this method we can concentrate the enzymes in the solution. Next one, stabilization of enzymes. Enzymes are sensitive to temperature, pH and also protease enzyme. From this condition we are stabilize it. How means adding stabilizers especially for glycerol and also sucrose. Next, we are controlling the temperature with the help of ice. Next, using some inhibitors to prevent the degradation of proteins. Next, factors affecting the isolation of enzyme. First one is source material. We are selecting the source material. The sources are first one is freshness and also enzyme abundance. Enzyme abundance is rich source. Enzyme rich source we can select it. Second one is pH and temperature must be optimal for enzyme stability. Enzymes optimum condition only which have the more activity. Then protease activity, protease means which acts on proteins which degrade it. So can degrade the enzyme if not controlled. Protease present means which degrade the proteins. Next, buffer composition, which helps the maintain enzyme activity. These are the factors whenever isolating the enzymes. Next, move on to purification of enzyme. First, we will say about introduction about enzyme purification. Enzyme purification is the process of isolating a specific enzyme from a complex mixture, then removing unwanted proteins and also contaminants. The goal is to obtain highly pure and active enzyme for industrial, medical and research applications. Next, the methods for purification of enzymes. Enzyme purification is based on solubility, size, polarity and binding affinity. The production scale, timeline and properties of enzymes should all be considered when choosing the proper separation method. Some of methods are centrifugation, ion exchange chromatography, gel permeation chromatography, electrophoresis, affinity chromatography, high performance liquid chromatography, salting in, salting out procedure, dialysis, lyophilization and crystallization. These are the technique we are using the purification of enzymes. First, we will see about centrifugation. Centrifugation is a mechanical process that utilizes an applied centrifugal force in field to separate the components of a mixture according to density and or particle size. Second one, ion exchange chromatography, which separates the proteins based on charge. Charge means either positive or negative charge. Positive or negative charged resins bind to protein of opposite charge, which allowing selective elution. Here we are separating positive protein means which binds to negative resin. Negative protein means which binds to positive resins. Remaining are passed out. Third one is gel filtration or size exclusion chromatography, which separates proteins based on molecular size. Here a smaller molecule enter into the column matrix. The larger molecules elute faster in between the gels. So the larger molecule moved faster than smaller molecules moved lately. Fourth one, electrophoresis and purity analysis. Here we are especially using SDS page. SDS means sodium dodecyl sulfate polyacrylamide gel electrophoresis, which determines the molecular weight and purity of enzyme. One more electrophoresis technique, isoelectric focusing, which separates the protein based on the isoelectric pointing. Isoelectric point means each proteins have the different isoelectric point. In this condition, the isoelectric point, the charges are 0. Next one is affinity chromatography, highly specific technique using ligand binding interactions. A ligand attached to column binds only the target enzyme, which is then eluted using competitive substrate. In this technique, separating solutions we are added top of the column. The specific proteins only bind to target resin, remaining are eluted out. Sixth one is high performance liquid chromatography, a new technology for purification of protein. In this technique, we are minute amount also separated and also purified. Next one is salting in, salting out. The solubility of proteins usually increases slightly in the presence of salt, which is referred as salting in. In this condition, small amount of salt present in the solution which increase the solubility. This is called salting in. However, at higher concentration of salt in the solubility of proteins drop sharply. The proteins can be precipitated out, which is referred as salting out. In this condition, in the presence of higher concentration of salt, the solubility will be dropped, so that the higher concentration of salt we can remove it, the solubility will be increased. So these conditions are called as salting out. Next one is dialysis. After purification, there may be some small molecules present in the enzymes. They can be removed by dialysis procedure. The dialysis is a separation technique that facilitates the removal of small unwanted compound from macromolecules in the solution by selective and phased diffusion through a semi-permeable membrane. Next one is lyophilization. After purification, the concentration of enzymes in the solution may be very low. In order to concentrate the enzymes, lyophilization techniques are used. Even ultrafiltration can be done to concentrate the sample of enzyme. Next, crystallization. The ideal way to complete a purification procedure is crystallization of enzyme. Enzyme crystallization is the process of forming a solid ordered crystalline structure from an enzyme solution. This process used for both protein purification and structural studies, which allowing researchers to isolate and analyze the enzyme structure. I hope, dear friends, do you understand about enzyme isolation and purification? Thank you all.